I'd like to hand over to Victoria and take us through her work on keratopathy.
Thank you. So, hi, for those of you who don't know me, my name is Victoria, and I work with Professor Julie Daniels at the UCL Institute of Ophthalmology, and we've been working on trying to develop a human model to study Aniridia Related Keratopathy, which is, of course, as you know, the opacification of the cornea. So the last time I was here, I spoke to you guys about how I was trying to make healthy corneal cells aniridic by trying to get rid of the PAC6 in these cells. And this is quite a tricky thing to do. So in the meantime, we've managed to set up a collaboration with Steve Tuft at Moorfields Eye Hospital, where we're able to get aniridic samples following operations where they've either had a scraping or a full corneal transplantation. So we can use these samples from the human patients to grow cells and to then study the effects and compare them to control cells and try to understand a bit more about what's going on in Aniridia-related keratopathy. So as Martin was saying, the cornea is made up of different cell layers, the outermost being the epithelium, which is what he just talked about. But I'm going to focus today on the corneal stroma, which makes up the majority of the thickness of the cornea. And the stroma is made up of specialised stromal cells and the collagen, the glass, as Martin was telling you about, that is transparent. And the arrangement of the cells and the collagen is highly organised in the stromal tissue. And this is essential to maintain transparency of the cornea. So a lot of studies have looked at possible things that are going wrong in Aniridia-related keratopathy. Starters, as Martin talked about, is abnormalities in the epithelium. And there's also complications with the wound healing response and an imbalance between wound healing and repair. And another thing that's also been found is that there's differences with the stroma and a thickened, more irregular stroma. So a lot of studies that have been carried out have been done on this mouse model, which is really useful for us to figure out what's happening with the cells in vivo or in a body. So even though quite a significant amount of work has been carried out on the epithelium, the outermost skin layer of the eye, there's also been indications that things are also going wrong in the stroma. So for example, I said it's being noticed that there's a thicker central stroma in the aniridic mouse cornea. There's also an irregular alignment of collagen in the stroma. And it's been found that there might be defects in these specialised stromal cells, which help organise the collagen to help maintain transparency. And also it's been found that there is an increase in stromal cell death following injury to the cornea. So because we can grow cells from these patient tissue that we get, the research aim was to grow the cells from this tissue and compare them to control cells and to investigate how or what differences we see. And this helps us get a better idea of how corneal opacification might progress. So these are examples of the corneal tissue that we receive. On the left, there's a control central corneal button, we call it. So this is a leftover piece of tissue once they have taken the bottom endothelial layer and a part of the stroma for transplantation for specific corneal problems. So the remainder top half of the tissue gets sent to us for research and we can grow cells from that tissue. And these control corneas that we get obviously have no problems with them, they're healthy, fully functioning corneas. And on the right we can see an aniridic cornea where the transparency is lost. You can see infiltration of blood vessels there, and so we can use these two types of tissue to grow cells from and compare any differences. So the way that we grow cells from this tissue is we use a specific enzyme, which is a bit like Pac-Man. It munches up things, so it munches up the collagen. And so we end up with the cells floating in a sort of liquid. So then we can extract the cells from what was a solid cornea and grow them in Petri dishes in the lab. And so one of the first things that we noticed about the cells is that the aniridic cells that we grew from aniridic tissue, they look different from the control cells. So in the aniridic stromal cells, we can see fine extensions growing out from the cells, which are pointed out by the white arrows. And so the fact that these cells look different to each other led us to believe that maybe they behave differently as well. So one way of looking at cell behaviour is to assess how fast they grow. So we take again the cells and we put the same number of cells into petri dishes, and each day we count the cells. And after the third day, we found that there's a significantly higher number of cells in the aniridic petri dishes. So the dark bar here is the aniridic stromal cell count, and the white bar is the control cell count. So they are growing at a significantly faster rate than the control cells, which might have some sort of effect in the overall scheme of things. And another way we can look at cell behaviour is to look at cell migration or cell movement. So as Martin was saying earlier, there's defects in the migration in the epithelium. So we also wanted to see whether the stromal cells have an effect, Aniridic has an effect on the stromal cells migration. And so how we do this is we take the cells again, we put them on this specialised membrane. And with this membrane has pores, tiny microscopic pores in it, which are small enough so that the cells can sit on top, but also cells can change shape and squeeze through remarkably small holes. So if they want to get somewhere, they will go through these holes, these pores. And on the other side of the membrane, we put what we call a chemoattractant. And this obviously attracts the cells. It's a bit like a dog is attracted to your plate of food. So we want to see if these cells are capable of migrating through these pores and onto the other side of the membrane. So we put these cells onto the membrane, and after 24 hours we can count how many cells are on the underside of the membrane, how many cells have moved and migrated. And what we found was the migration of the aniridic stromal cells is significantly impaired compared to the control cells that we've been monitoring. So again, it seems like there is some sort of effect on migration in the corneal stroma. So the data I've shown you so far is mostly work in 2D, whereas obviously cells in your body are in 3D. So we want to try and recreate a three-dimensional environment. So even though we've taken the cells out of the cornea, out of its 3D environment, so that we have lots of cells that we can use to have lots of different experiments, whereas if we just kept the cornea as it was, we'd have very little to be able to do with it. So we make a gel that we can put these cells in, and it kind of mimics the 3D environment that it should be in. So we put the cells inside a liquid collagen, and this, we're able to set this collagen much like you would set jelly, if you're going to have jelly for pudding. And so this forms a nice 3D environment for the cells. It's not exactly as you would have it in vivo, but it's a good start. So when we put these cells in 3D, we again want to look at how they behave and how they look. And we found that when we observed the cells, they looked different. So they have a much different shape than in control cells compared to aniridic stromal cells. So we then sort of do some measurements of the cells, and we put these measurements into an equation, which I don't even understand sometimes, and then it plugs out this circularity value. So circularity, as it sounds, is a measurement of how round or how circular a cell is. So a higher circularity value is obviously more circular, and we found that the aniridic stromal cells are significantly more circular than control cells. And in fact, when you look at the control cells, so on the picture on the left is the control cells, and these cells formed nicely elongated shapes, and they're all aligned in parallel with each other. Whereas the aniridic stromal cells on the right, as you can see, they're more round, less elongated, and they don't align with each other. So not only is the shape different in 3D, but they also seem to be not organising themselves in the same way as the control cells would. So in summary, these stromal cells that we've grown from aniridic tissue, they look different, they grow faster, they migrate slower, They have a different shape in 3D, and they don't align or organise themselves in the same way as control do in 3D. So this may suggest a possible involvement for this corneal stroma in arc progression. So what does this mean? So Aniridics stromal cells display these properties that might contribute to corneal pacification. This gives us more of an idea of an understanding of what's happening, which would help determine better therapeutic approaches in the future. So that's it. Thank you guys for listening. Thank you also to Shine the Light on Aniridia, who's sponsored a lot of the work that I've shown you. And also we managed to raise some money from the Special Trustees in Moorfields Eye Hospital as well. Thanks very much.